Welcome, students, to Module 6, where we begin to talk about the crime scene investigation, and one of the most important variables when a corpse or decedent is discovered, which is the postmortem interval. So when you have the discovery of a crime scene, you also have discovery and reveal of evidence that is contained within the crime scene. And of course, when there is foul play involved and the death of an individual, either by homicide or suicide and what have you, then you also have to determine the postmortem interval. The postmortem interval is one of the most important variables because it tells us post, meaning after death, and then the interval between when the individual died and then when other individuals, that is maybe an informant, somebody who discovers the body, or, you know, the law enforcement themselves or the crime scene unit actually encounters that individual, the corpse of that individual. It's very important because if you look at even the television show 48 Hours, the premise of that being that if the detectives don't have a lead within 48 hours, the case may go cold. And so the postmortem interval is very important because it will tell them how long that individual has and their body has been there, and that can be instrumental in solving a case. And so in this lecture, we'll cover crime scene discovery and then some of the key stages of mortis. There are more than just three, what have you, but stages of mortis, including decomposition, as well as forensic entomology, can actually inform on the postmortem interval. Crime scene discovery can occur in a number of different ways, and they're not mutually exclusive. You can get an anonymous tip, maybe a law enforcement informant. You can have an individual who hears about a missing person report and then kind of scours some area or geographic space and happens on some or finds some type of evidence and then calls that in. You can have construction or land development. You can have, I've heard stories of individuals who buy a home and then start renovating it, either the interior or maybe, you know, concrete driveway or something on the outside, and then they discover the corpse of an individual. You can use local or historic knowledge. It can be chance or luck, but for the most part, there has to be some type of field work at the scene, at the scene of the crime, or at the scene of disposal of the corpse or the body or the human remains. Sometimes there are sophisticated techniques that are used. You will use very simple surveys, which will be like a systematic search of the landscape for evidence. And you'll see these types of line searches all the time, where maybe some article of clothing or a wallet or an ID is found in a forest or a field, or there are reports from an informant or anonymous tip. And it can be law enforcement officials, it can be volunteers that will get together and then conduct a line search to try to find any other evidence that is contained in that geographic space. And this is where you'll have people that will line up parallel to each other, and then they will walk across this space like this image, and then they'll walk and then maybe pause every so meters or feet in an interval to try to look around and find anything that can also lead to evidence collection or more clues for law enforcement. But you can also have not just basic surveys, but you can have more sophisticated techniques like ground penetrating radar or electrical resistivity. And what these will do is will send signals into the ground and will also give us, information if there are disturbances in the soil. Oftentimes it can locate, a dead corpse, of an individual. And after, other, evidence might be found after a basic survey, then there's documentation of the surface. You might be finding areas of disturbance because oftentimes you'll see individuals will maybe bury the corpse or decedent, and this will cause disturbances in the soil that are visible, and then they will locate typically the focal point of the crime scene. And so what may also happen is, as you're doing a survey, it might be done systematically, and then they start with a line search, and then they just start finding bits and pieces of evidence. Or when law enforcement locate even just one piece of evidence, then they may do a radiating search around that. But oftentimes that's what they're looking for eventually, is the focal point of the crime scene, whether that be bits and pieces of evidence. The focal point oftentimes, if a decedent or the corpse of an individual is found, that is oftentimes the focal point of a crime scene. One of the first stages of mortis, and particularly that will aid in, discovering and revealing the post-mortem interval, right? How long it has been, or time that has passed since individuals encounter a corpse or the body of a deceased individual. And when we look at the internal body temperature of humans, generally that is around 98.6 degrees. Of course, that can fluctuate to 97 or 99, but generally anywhere, and most people will use centigrade or Celsius here, and other people use things like Kelvin, because those are oftentimes universal ones that are used in chemistry and forensic science. So 37 degrees Celsius or 36 degrees Celsius is the general temperature gauge that's used to measure the internal body temperature of an individual. And once an organism like a human dies, then that body temperature will decrease, because we're homeothermic, meaning that we regulate our own body temperature as opposed to a cold-blooded organism, right? So that's the homeothermic is the technical term for warm-blooded. And so when a human dies, then the body's temperature decreases until it reaches the ambient temperature. That is the temperature that is where that organism became, or that individual, a human, became deceased. So when a human dies, that internal temperature decreases 2 degrees Celsius in the first hour, and then 1 degree Celsius each hour until it reaches that ambient temperature. Now, what I have here is a general chart that shows the estimation of time since death, or the postmortem interval. And you have stages like algor mortis, rigor mortis, and liver mortis. Each one of these we will go over and discuss. And it is, algor mortis being one of the first ways to kind of determine this, because it usually is instituted first, and then things like rigor mortis and liver mortis will persist, and we'll discuss those. But understand that, something like temperature change or temperature loss in a corpse, algor mortis, that's something that is dependent on the environment. The environment that, the corpse and the decedent are exposed to. And so you can think about an environment which is, our hot summers. You may actually have a corpse that it has increased in temperature. You may see a greater loss in temperature if an individual dies in a colder environment. So we have researchers all over the world in forensic science that are studying this phenomenon, and it's different in different environments. So this is something that I want you to begin to try to understand throughout the course, is that the techniques that we have in forensic science and forensic anthropology, they're oftentimes dependent on the environment that the individual is subject to foul play or dies in. And, this is important because our standards need to be for those localized environment, and oftentimes standards that are for, worldwide application are not sufficient. The next stage that sets in that also coincides with algor mortis is livor mortis, and that is the setting or pooling of blood in a corpse. The image you have here shows an individual's torso and lower abdomen and rear region. This individual is lying on their left lateral side, and it is exposing in oblique view the right lateral side as well as the posterior or dorsal aspect. Livor mortis and the pooling and settling of blood usually begins at about 1 to 2 hours after death. It is well developed about 3 to 4 hours after death, and then maximum lividity is usually about 8 to 12 hours after death, and then maximum lividity will be fixed from that point onward until you have advanced decomposition, meaning that once you have the maximum lividity, it is retained for a number of days until you reach more, more advanced stages of decomposition. And so livor mortis can be a pretty good indicator of the postmortem interval, particularly if you're encountering a corpse a few hours after death and it coincides with algor mortis. But then after about 12 hours after death, livor mortis alone is not a very good indicator of the postmortem interval. You can also notice that livor mortis will also be dependent on, what condition and the placement of the corpse when it is actually discovered at a crime scene. It also is dependent on the number of areas, including, those that have lots of adipocere or adipose tissue, right? That's fat tissue. And so you can see on this individual, the buttocks is an area where there's typically lots of fat mass or fat containment, adipocere. And so blood will take more time to pool in those areas, or it may not pool in a way that's visible. So you can notice that areas that have less, fat mass will actually present more visibly on the epidermis or the skin. Understand that those factors are also implicit when we look at livor mortis, and it's not like the whole corpse completely turns red. It, again, is dependent on the side the individual is actually on. And matter of fact, this individual was discovered on the right side. This individual's corpse was just rotated on the left lateral side so that it could be viewed and recorded for this photo documentation. So just be aware that it's not something where, it's ubiquitous all over the body. You'll see bits and pieces of variation dependent on placement of the body or the corpse, when it was discovered, before where it's placed and then actually encountered. And then it also is dependent on body composition. Rigor mortis is yet another stage that can help us with the postmortem interval. And this is effectively body stiffness that occurs following the death of an individual, and it's because of a number of different chemical changes and release in muscle tissue. And it typically occurs 1 to 2 hours after death, and then you will have maximum stiffness after about 12 hours, and then this stiffness will gradually decrease in the next 24 to 48 hours. So what you'll see is an individual will begin to get stiff, and it will be incremental, right? Slowly and surely, stiffer and stiffer. And then after those chemicals are released and expended, then the stiffness will begin to reduce again. So something very important to know about rigor mortis is if you see a little bit of stiffness, is it leading to more body stiffness, or is it leading to reduced body stiffness? After about 48 hours, the body or the corpse should not be stiff anymore. And so that would mean rigor mortis really isn't very good past 48 hours. Decomposition is the process by which the body begins to become depleted in terms of the integrity of its tissues. And this is oftentimes brought up by the elements, but it's also brought up by microorganisms. Decomposition usually occurs after about 12 to 18 hours. And then it will intensify, and then you'll have different stages of decomposition after all the soft tissue is gone. And then you'll get to things like skeletal decomposition, where you'll have weathering of the bone, as well as other organisms like rodents. They will gnaw on bones because their incisor teeth grow continually, and they need something to trim them. So you'll see those marks once you even get to skeletal decomposition and weathering the bone on the outside. Autolysis is the first stage that typically occurs in decomposition at about 12 to 18 hours and will persist into things like putrefaction. But autolysis is where you actually have the digestive enzymes that are in your mouth, that are in the lining of your esophagus and into your stomach, and a number of other different enzymes and reactions that are occurring all over your body. They're used to breaking down tissues. They're used to doing this to maintain your body. They're doing it to break down your foodstuffs that you're intaking. And then, for these enzymes to persist, they need to feed on something. And so if they're not getting anything else, because there's no uptake during death, then those digestive enzymes begin feeding on your body tissues. And then after those enzymes have fed enough on body tissues and actually will eventually die off, then you will have other bacteria that will begin feeding on body tissues. Understand that you have a number of different bacteria and fungi that are living on your skin, right? And other small mites and stuff that are living on your skin and your hairs right now on your body. And they create a balance on your epidermis and skin, for instance. One of them gets a little too out of control, then you have things like acne or psoriasis or staph infection, Staphylococcus aureus. You also have a number of different bacteria that live in your stomach. Good bacteria. These are things like lactobacillus, right? Things that you will ingest even when you take yogurt or maybe something like kombucha, which has lots of different live active cultures in it. And those help create a balance in your stomach, particularly in Western diets that consume a lot of sugar. But those bacteria are used to feeding on things that, again, that you are intaking, as well as maintenance on your skin to create this type of balance. And so they no longer have something to feed on, and then they start actually feeding on your body tissues. And this type of decomposition is exclusive to soft tissue decomposition. It does not necessarily affect the body once it becomes completely skeletal. Forensic entomology is very useful in determining or estimating the postmortem interval, and that is looking at the insect life cycle of particular insects. And there are a number of different insects that begin using the corpse for feeding, but also things like reproduction. And one of those insects, and matter of fact, the first insect that actually begins using the corpse, utilizing it for its purposes of reproduction, is the blowfly, or the common housefly. And so when you encounter a corpse, what will typically occur is that you will begin to see eggs being laid by flies. And from when the body or the individual has died, from about 23 hours or so, almost a full day, you will have eggs then move into the larval stage 1. And these can be measured to determine different larval stages. And then from about 24 hours to 27 hours, they will persist in the first larval stage, and then they will make it to larval stage 2 after roughly 50 hours. And then from 50 to up to about 72 hours, they will maintain in larval stage 2, and then after 72 hours, they will move into larval stage 3. And then from larval stage 3, they will move into the pupal stage. So from 72 hours to about 202 hours, they will be in larval stage 3, and then they will reach the pupal stage. And then after the pupal stage, after 143 hours have passed in the pupal stage, you will reach, an adult fly. So what entomologists can do, and, entomology has been a booming field in forensic science for these reasons, as well as the diversity of the types of insects that are found, colonizing this corpse. It can also be helpful in the postmortem interval. Other different, other types of insects that are found in specific environments also will be good indicators of the postmortem interval. Now you'll see that this life cycle is, determined at 70 degrees Fahrenheit. What is it like at 90 degrees Fahrenheit? 100 degrees Fahrenheit? What is it like when it's less than 70 degrees? So understand that, again, this is something that you'll likely see slight variation in different temperature intervals. So this may not be a hard or fast rule in all environments. So that's something to be aware of, just like when we look at something like algar mortis and those calibrations. Lastly, when we look at concepts in forensic entomology, insect succession can be a really good indicator of the postmortem interval. When you have the blowfly, the first, insect that begins colonizing the corpse, after, a number of days, you're going to see insects of a blowfly in a number of different stages in their life cycle, because there are a number of different adult flies that are starting to lay eggs, and they are done at different intervals. So if you happen on a corpse and they only have eggs, then you're pretty sure that you're in that first 23-hour window. Now, if they only have one type of larval stage or pupal stage, that's fairly uncommon. You're likely going to see after that 24-hour interval, eggs and larva. So and possibly in different stages, and then after 5 days, then you start seeing other types of insect, like a flesh fly. A few days after that, a cheese fly. After 2 weeks, you see a coffin fly. And then after about 2.5 weeks, then you see a skin beetle. All right, there are types of dermestid beetles that actually will feed on flesh and even bore holes through bone. And so the observation of different insect organisms will tell you, or have a really good indicator of the postmortem interval. And you'll have to do this in unison. And of course, you're going to want to use concepts of all of these, right? Of the different stages of mortis, of the different stages of decomposition, of the different stages of insect life cycles, as well as their succession. All of them, as much information that can be used at the crime scene when you discover a human body that is an individual is deceased, or a corpse. All of them should be used in unison. Now, there are some that might not be useful at certain stages. And so you would want to prioritize, of course, the ones that are most useful, but you should definitely use multiple methods in determining the postmortem interval, if at all possible.